Assalamu alaikum students. Today we will study about the cell and cell division. But before watching this video, please subscribe the channel.
Assalamu alaikum. Today we are going to study about the cells and the cell division. Cells are of two types, body cells or somatic cells, which are having 2n or diploid number of chromosomes. The other type of cells are sex cells or germ cells or the gametes, which are having n number or haploid number of chromosomes. Haploid means half the number of chromosomes because these cells are the gametes, and the male and female gametes each has the haploid number of chromosomes in their nuclei. So when their nuclei fuse at the time of fertilization, the normal number of chromosomes is restored. The new cells come when the pre-existing cells, they divide. The cell division is of two types, mitosis, in which one body cells with diploid number of chromosomes divides into two cells which are also having diploid number of chromosomes in their nuclei. The other type of cell division is meiosis, and meiosis takes place in the body cells of the gonads to form the gametes. One body cell with diploid number of chromosomes divides into 4 cells which are having half the number of chromosomes in their nuclei. The cells are not always in their dividing phase. The phase when the cells are not dividing, that is called the resting phase or the interphase. The diagram you see, the yellow part of the diagram shows the interphase. It is that phase when the cell is not dividing. And you see that this is the phase when the mitosis is taking place. Mitosis is when the nuclear division of the cell is taking place. And after that, there is the phase which is represented with the name cytokinesis. That means the cytoplasmic division. The diagram shows the hereditary material in a cell when it is in the interphase or non-dividing phase. The hereditary material appears in the form of threads called the chromatin threads. These chromatin threads, they are made up of DNA molecule, and this is structure of one of the chromatin thread. It has the central part called centromere, and this is the chromatin thread. Just before cell division in the interphase, the replication of chromatin thread or DNA takes place, and exact copy of the DNA is made, or the replica of DNA is made, which are called the sister chromatids. These chromatin threads, they coil and condense and they get shorter, and this is the shape of the chromosome. Chromosomes means the condensed form of the chromatin thread. If it is a human cell, then there will be 46 number of chromosomes in the nucleus of the cell when the cell is in the dividing phase. Overview of mitosis. Parent cell with diploid number of chromosomes. DNA replicates, that is, the copy of DNA is produced and the chromosomes appear in their actual shape, that is the X shape. And then this parent cell, after undergoing these 4 phases, that is prophase, metaphase, anaphase and telophase, it is divided into 2 daughter cells with diploid number of chromosomes in their nuclei. Phases of mitosis. The prophase has been divided into early prophase and late prophase. In early prophase, we see that the chromosomes are formed as the DNA replicates. Centriole divides, and the 2 centrioles, they move towards the opposite sides. The ester or the star shape appears around each centriole, and the spindle fibers are also being made. In the late prophase, we see that the nucleolus and the nuclear envelope or the nuclear membrane disappear. Metaphase of mitosis, we see that the spindle has been made. Spindle is basically the shape which is broader in the center and it is narrow or tapering at the ends. The ends of the spindle are called the poles. There are two poles in the spindle. These are the poles. These are the two poles of the spindle, and this center part which is broader, that is called the equator of the spindle. During metaphase, the chromosomes, they arrange themselves on the equator of the spindle in a single file or in a line. After that comes the anaphase. During anaphase, the centromere, which is the central part of the chromosome, centromere divides and the chromatids of the chromosomes, these are the chromatids, they get free. The spindle fiber gets shortened and they contract and they move the chromatids towards the opposite poles of the spindle. When the chromosomes, they reach to the opposite poles, then the anaphase finishes and the next phase starts, which is called the telophase. In the telophase, the nuclear membrane reappears because the chromosomes, they have reached towards the poles. The spindle fiber disappear, nuclear membrane reappears, nucleolus reappear, and this is called karyokinesis, that is the division of nucleus. After that, cytokinesis, that is the cleavage of the cell membrane takes place and the cytoplasm divides, and in this way the two daughter cells with diploid number of chromosomes are produced and the mitosis finishes. Mitosis in plants is similar to animal cell except there are no centrioles in the plant cell and the spindle fibers are made without centrioles and there is no cleavage of cytoplasm at the time of cytokinesis, but between the two daughter nuclei of the plant cell, the vesicles fuse to form a cell plate. These vesicles are actually the fluid-filled spaces formed by the Golgi apparatus. The cell plate made by the vesicles eventually makes the cell wall between the two plant cells.